Hello students, today we continue with 11.3 and we are still looking at the kidney. Today we're going to be looking at osmoregulation, and remember that this is the third process that happens within the nephron, and it happens within the loop of Henle. Alright, so let's move to that topic. Here we go. So the essential understanding is that the loop of Henle maintains hypertonic conditions in the medulla. So if you remember, hypertonic means that it has a higher solute concentration and a lower solvent concentration. So less water, more solutes. And in this particular case, the solutes happen to be salts. So we know that osmoregulation controls the water balance of the blood, tissue, or cytoplasm in a living organism. It happens in the medulla of the kidney, and remember that this is the inner portion of the kidney, and it involves two events. We're going to be looking at these two events. The first event is that the loop of Henle establishes a salt gradient, meaning that the medulla is going to become hypertonic, and that's important. And the second event is that antidiuretic hormone, ADH, regulates the level of water reabsorption in the collecting duct. So we're going to be looking at these two events separately. So let's start with the first event, which is establishing a salt gradient. So the first thing we need to understand is the structure of the loop of Henle. So remember that this right here is the whole nephron, right? And this portion right here is the loop of Henle, right? Now, remember that the loop of Henle is surrounded by the vasa recta, which is the branches that stem off the efferent arteriole. So we see all of these capillaries surrounding the loop of Henle. So the function of the loop of Henle is to create a high solute or hypertonic concentration. So when we think of the differences between the medulla of the kidney and the cortex of the kidney, we think of the medulla as always having high salt and low water. And we think of the cortex of the kidney as having low salt and high water. So this picture right here is really helpful. It shows us the filtrate as it moves down the loop of Henle. So remember I told you that the filtrate, which is the urine, right, is going to be shown to you in yellow. And then it shows you in red the vasa recta, or the capillary that's surrounding the loop of Henle. Now, one thing to note about these two is that they happen to be moving in opposite direction. So this is what we call a counter-current exchange. And this is going to allow for the water to move into the capillaries and for the salts to also move into the capillaries. So we have the descending limb of the loop of Henle. And this descending limb, notice that it is permeable to water. So that means that water can exit the descending limb, and water then goes into the medulla part. The ascending limb, and this is this part, the ascending limb of the loop of Henle, this is permeable to salts. So sodium chloride, right? Salts. So these ions can permeate, leave the ascending limb of the loop of Henle, and they then go into this interstitial fluid area of the medulla. So that means that the medulla becomes salty, the medulla becomes hypertonic with low water and high salt. Okay? So what does that mean? That means that if you look at this picture right here, you can see the osmotic gradient that is being established in the medulla here. So this is the cortex region of the kidney. This is the medulla region of the kidney. And notice how, because of what happens here in the loop of Henle, this area becomes hypertonic. Okay, so notice that the osmotic gradient is increasing as you dig deeper into the medulla. Okay, so this is going to be important because as the filtrate moves up from the loop of Henle into the distal convoluted tubule and then into the collecting duct, right? The collecting duct is going to carry water from different nephrons, right? And from this particular nephron itself. So this water, when it reaches this area right here that is very hypertonic, right? Then knowing the properties of water, what is water going to do? This water that is found here in the collecting duct, this water is attracted to areas of high solute concentration. So then that means that the water within the collecting duct is going to leave the collecting duct, right? And where is it going to go? This is what we're going to talk about in just a moment, okay? So then that means that the more water that leaves the collecting duct, the more concentrated the urine is going to be, okay? So we don't want to dehydrate when we urinate, so this process is very important, right? We want to make sure that our urine is proportionate to the hydration status of our body. So if we are dehydrated, then that means that we need to release more water from this collecting duct, and that water is going to go back into our blood so we don't dehydrate. If we're perfectly hydrated, then that means that the water that is found in the collecting duct doesn't have to come out as much, and then your urine is going to be more dilute. Okay, so the second event that happens within the coordination between the loop of Henle and the collecting duct is that Adhd controls reabsorption of water in the collecting duct. So just quickly, if you remember from the diagram here showing you the structure of the nephron, remember that osmoregulation happens here in the loop of Henle, but it also happens here in the collecting duct. So we just finished looking at osmoregulation and how that salt gradient is established within the loop of Henle. So now we're going to move on and we're going to talk about water retention as it happens within the collecting duct and with the help of ADH, Antidiuretic Hormone. And this is another form of osmoregulation. So now we're going to be looking at the collecting duct region. All right, so let's go back to the slide here. So that's the second event. Adhd controls reabsorption of water in the collecting duct. So here you have a diagram that is showing you the blood vessel. It's showing you the tubule cells, which are the cells that are on the exterior of the collecting duct, and then the collecting duct lumen. So then that means that in the lumen of the collecting duct, that's where water is going to be present. Okay? So we've already said that the medulla is hypertonic, right? And this happened through the processes that we just saw in the loop of Henle. Now, the amount of water that's going to be released from the collecting ducts back into your blood system, back into your body, is controlled by ADHD, this antidiuretic hormone. Now, where does ADHD come from? ADHD comes from the posterior pituitary gland. And the posterior pituitary, once thought to be the master gland, we now know that it's not, right? Because the pituitary gland does what the hypothalamus gland says. So the pituitary gland is going to release ADH as a result of the hypothalamus osmoreceptors detecting that a person is dehydrated. Okay? So as the brain releases ADHD, right, into your bloodstream, ADH ADH is going to increase the permeability of the collecting duct to water. And that means that there is going to be a regulation of these aquaporins, which are these water channels that are found within these tubule cells. Okay? So what does that mean? Well, if a person is hydrated, then that means that your pituitary gland is going to release low amounts of ADH. So if we have low amounts of water, right, then that means that you're going to have low amounts of ADH, right? So if you have low amounts of ADH, that means that this water that is found in the collecting duct lumen is not going to leave at your body as much, right? Because the production of the aquaporins, or the regulation of the aquaporins, is going to be limited. So if a person is hydrated, there's low ADH, that means that little water is going to get reabsorbed, right? Because only a few aquaporins are opened. So less water reabsorbed back into your blood. What does that mean? Well, that means that the urine here that is being released of a person who is hydrated, that urine is going to be more dilute. Okay, so let's write that down. More dilute urine. All right, so what happens if a person is dehydrated, though? If a person is dehydrated, then the hypothalamus is going to detect that. It's going to tell your posterior pituitary to send high amounts of ADH. So ADH is going to be high. Well, that means then that ADH acting upon these aquaporins is going to open many of these aquaporins, and that means that the blood that is found within the filtrate of the urine, right, the filtrate, that is going to be taken back up or be reabsorbed more into your blood. So then that means that more water gets reabsorbed. So more water gets reabsorbed back into your body. And of course that makes sense because you're dehydrated. So you don't want to lose more water via your urine. So then that means that your urine becomes more concentrated. So a person who is dehydrated, since the urine is more concentrated, that urine is going to look darker because it has less water. A person who is normally hydrated, the urine, since it's more dilute, the urine is going to look lighter. Okay? So that is pretty much it for osmoregulation in 11.3.